I'm very happy to be here. Today I'm going to be telling you about a kind of bipartite story because there's a first part on what could be considered aneuploidy, and so chromosome gain or losses, whole chromosome gain or losses, and a second part on polyploidy, so the gain of the entire chromosome set. Before I go into our work, I should put this on. And so the next chromosome instability lecture series will take place in October 17. Please take notice that this has been changed. Initially, this was programmed for September. And so I want to just put this here and thank once more Aurélie, Isidro, and Florian for organizing this very nice chromosomal instability series. So to start with, today I'm going to be telling you about the brain. This is a picture of a human brain, which I like to describe as a really complex organ. I think people who work on different organs will always think that the organ they work on is complex. But this one is particularly complex because it really defines what we all are in the way we behave. And because when it's set up, when the brain starts to be developed, it has to coordinate proliferation and differentiation with, I would say, very little plasticity compared to other organs. But the most important aspect related to our work, and that's why I'm spending some time here with this slide, is that most brain development actually takes place during embryonic development, which is not the case of other organs. There are regions in the brain that also undergo neurogenesis in the adult, but the cortex, which is the part I'm actually interested in, it's set up and developed during embryogenesis. Now, the mouse brain, by comparison, is much smaller. Still, I would say that everything here is valid for the mouse brain. And the fly brain, which we'll also work on it's also complex, I would say, and it's basically ruled by the same type of principles, but I'm not going to be talking about fly brain development today. And the main reason for it's because what I have been having some message coming on. I'll just turn it off. And the main reason for it that I'm focusing on human and mouse brain, it's because they have very similar patterns in terms of organization. So here you can see a schematic diagram of the mouse and the human brain, and the part that we are interested here is this region that is called the ventricular zone, where if we zoom in, we can actually see what we call apical radioglial cells, which are the progenitor cells that are responsible to generate all the cells that will make the cortex. And these apical radioglial cells are bipolarly attached to the apical side, which is in contact with the ventricle, and to the basal side. And when they divide, they can actually generate other radioglial cells or some intermediate progenitors, or even more differentiated cells by direct neurogenesis, which I'm really not going to be talking about today. So the entry point to be actually studying the brain comes from this knowledge that there is this human disease called primary recessive microcephaly, or MCPH. This is a neurodevelopmental disorder that's considered to be rare because the frequency is between 1 in 50,000 to 1 in 100,000. It's characterized by reduced brain size at birth, normal overall brain structure and organization. And what is interesting is the overall body size and the size of other organs remains unaffected in these patients. The price of having a smaller brain, of course, comes with having moderate to severe intellectual disability and epilepsy. And what is interesting from a centrosome point of view, which is where our entry point is for aneuploidy or even polyploidy, it's related with the fact that mutations in genes encoding mitotic proteins, among those many centrosomal proteins, have been identified in MCPH. One of them is ASP-M, which is a microtubule binding protein, and it's actually the most frequently mutated gene in MCPH. And ASP-M has several domains, such as calpain domains and isoleucine glutamine repeats, IQ repeats. I'm not going to be talking about those, but what is just important to keep in mind is that ASP localizes to the spindle poles, and it stays to the spindle poles until metaphase, as you can see here in pictures taken from human cells from the Akhmanova lab. Now, a few years ago, my lab characterized some existing mutations on ASP-M flies. They were actually 40 years old. They come from work done by Pedro Ripoll and David Glover and Caetano Gonzalez, where we actually realized that indeed flies that are mutant for ASP, which is a homologue of ASP-M, have brain size reduction. And what was interesting is any of these mutations could be rescued with just one copy of full-length ASP, as you can see over here. Now, surprisingly, the Huttner lab has generated mouse models for with ASP-M knockouts, and in these, the microcephaly was relatively modest, as you can see over here. But more importantly, the Walsh lab has generated a ferret model for ASP-M loss, and in here, the microcephaly phenotype was actually quite important. Now, our work in flies have shown that ASP mutations result in chromosome segregation defects, as you can see over here, and micronuclei, and we put forward the possibility that maybe defects in chromosome segregation will be at the basis of microcephaly. Now, this is highly debatable, and it was kind of still, and it's still actually argue a lot for and against, because aneuploidy and chromosome segregation and micronuclei are normally associated with cancer. There are even hallmarks of cancer. And so it was difficult to conceive that the human brain could actually have cells with this type of chromosomes. And so one of the questions that we are interested in is to understand what are the underlying mechanisms leading to 50% brain size reduction in ASP-M patients. And so this has been actually the work of someone called Sandrine Pasmar, an MD PhD, who came to do a sabbatical in our lab at the time with the purpose of studying mitosis and understanding how mitotic spindles can be characterized. And since then, with her team, with her colleagues here, in this picture, Sandrine has developed very interesting tools to study ASP-M function in the human brain. So using patient-derived in pluripotent stem cells, neural induction can be induced, which will lead to this formation of these beautiful rosettes that look like this, where you have centrosomes towards the apical side, with the mitotic spindles nearby. It's not like a ventricle, not like a brain, but there are some aspects of organization that we can identify very easily. And Sandrine has, she's a neuropediatrician. She works with a lot of these families that have children with primary recessive microcephaly, and she has characterized some rosettes derived from different ASP-M families, and here I'm presenting two. One that contains mutation on quite early in the protein, as you can see, exon 3, and the other one towards the end on exon 24. And in any of those, we cannot detect ASP-M from fibroblasts taken from this patient. Now, since we want to understand if there is no contribution of the genetic background to the phenotypes that I'm going to be describing you. I'm one of the important things to do is to correct the mutations, which has been done for patient 1 on CRISPR-Cas9 technology and obtain an heterozygous rescue, where for patient 2 this was actually not possible, and so the mutation has been generated de novo in iPSs from healthy donors, and what we call healthy controls. And so here are rosettes from ASPM patients. You can see that they are much more disorganized as the one I just shown you before, but more importantly, you can certainly identify very abnormal spindles like the ones you're seeing here on the right, which are basically multipolar and with the quantifications over here. More importantly, we also identified bipolar spindles with chromosomes missegregating, as you can see here, in ASPM patients and also in the de novo generated rosette. Now, we wanted to know what will be the fate of these aneuploid radioglial cells, and so past work from our lab has shown that in Drosophila, aneuploid neurostem cells will undergo premature differentiation, where in mice, they will undergo apoptosis. So Sandrine characterized the behavior of intermediate progenitors, here revealed by TBR2, and apical radioglial progenitors by SOX2, and we can see that there is a significant decrease in the number of SOX2 positive cells in these rosettes when compared to controls, and an increase in the number of TBR2, at least in some of these clones, suggesting that indeed some of these rosettes were undergoing premature differentiation. In addition to that, we also see an increase in cell death revealed by CLISCAS phase 3, suggesting that more cells are also dying in patient-derived rosettes. So with this, we learned that SPM patients-derived rosettes show multipolar spindles, chromosome missegregation, premature differentiation, and increased level of apoptosis. The key question, of course, is what will be the extent of these results that can actually be also detected in vivo. And so Sandrine actually took the challenge of now working with fetal brain, and these are actually massive tissue structures that we can use dedicated mosaic tissue stitching to basically take at low magnification fields of view of both control and ASPM fetal brains, and then at higher magnification tissues, by identifying the zone, the region where we are in the brain, we can characterize mitosis. And it was actually obvious that if we focus on ASPM brains, these are taken from 22 gestational week, we can see very abnormal configurations, not only of anaphases that we don't even know how to actually describe them, they look very abnormal, maybe multipolar, but also with bipolar metaphases where we can see chromosomes localized at the poles and also not congressed at the metaphase plate. The surprise came for also in controls. We can see what looks like a perfect metaphase plate that indeed has a polar chromosome associated with this NUMA positive pole. And so even if these missegregation events or these misbehavior events in the control brain remain very sporadic, as you can see over here, and infrequent, this is telling us that these can actually be captured when we're looking at the human developing brain. And so we are building this kind of model where we think that defects in mitotic spindle assembly, at least in ASPM, can lead to defects in chromosome segregation in aneuploidy, which will trigger either premature differentiation or apoptosis, leading to a depletion of the population of apical radioglia and to a smaller brain. And the key point is that this low frequencies of chromosome misbehavior in the human control brain is also teaching us or telling us, informing us that we need to understand why these cells are actually generating this type of defect. And so this brings me to the question that we want to address now, which is how to explain the mitotic vulnerability of neurostem cells. So this part of the project will be developed in mouse. And what I need to tell you is of some of our past work where we compare early neurogenesis in the mouse brain with late neurogenesis. And I'm going to be using early and late throughout the talk. And this actually refer to standpoints in development of embryonic day E13 and a half and E16 and a half. And what we have shown is that apical radioglial cells, so these cells that I tell you that are bipolarly attached, have mitotic spindles with high levels of astromicrotubules at early developmental stages and high levels of chromosome segregation. For those of you who do not know what astromicrotubules are, the microtubules that actually emanate from the poles and contact the cortex. And we do not think that this is just correlative because we have shown that if we modify spindle morphology, then the frequency of chromosome segregation actually decreases. And so taking into consideration that we see this different power capacity in generating abnormal chromosome segregation, we wanted to ask the question which factors can actually lead to these differences in chromosome segregation accuracy in early neurogenesis. And this work is spearhead by Véronique Martiens in some research in my lab, with a lot of help from Lynn Jawish and Margot Malos. And so what Vero did was to first characterize E13 and E16 brains to have an idea of how these apical radioglial cells, so the progenitors of the brain, are actually behaving in these two different developmental stages. And she could see that there is this high density of ARGs at early stages that is decreased at later stages. And also this almost evident, if you think about it, higher surface area at later stages, because of course at later stages now cells have started to undergo asymmetric divisions so that they can generate progenitors that or differentiated cells that leave the ventricle area, and so there's more space for these cells to actually occupy this ventricular area. If Vero measured volume, she could see that this was also different because individually each cell has a higher volume when you look at these later developmental stages compared to early stages, volume here, and their aspect ratio also quite different. And so we wanted to know if this was cell autonomous or not. In other words, can the crowding that we are seeing influence the way the mitotic spindle and the chromosomes are being segregated in the mouse brain? Now, we could not do this in vivo if we wanted to know if it's cell autonomous or not, or at least not in an easy way in mice. And so what we decided to do is to invest in actually being able to have primary cultures of embryonic ARGs, which Vero manages to obtain by dissecting cerebral cortices. And what she can do is to obtain embryonic brains from any developmental stage and treat them in the way to obtain ARGs that then are cultured in vitro. And with the help of Karine Vignard, we actually can put these cells under physical constraints of different sizes. These are coated with fibronectin, and then we can also mimic cell-cell adhesion with NCAB-FC. And if you look at these graphs, we can see this different mitotic aspect ratio as expected according to the constraint. But more importantly is that increased constraints respond with an increased number of astromicrotubules and also on their length. Now, unfortunately, we cannot film in this, sorry, in this kind of PDMS wells. And so if we want to see how chromosomes being segregate, this is a problem. And so we decided to actually develop another system where the same cultures are um, um, set up in different ways, either as 3D floating aggregates or as 2D adherent cultures on fibronectin. So here I want to really stress out the important point that we have embryos, Vero dissects their brain and puts in culture these cells in a way that it's favoring ARG proliferation. The same culture, exactly the same culture coming from exactly the same animals, are then put into these different modes of culture so that in 3D they will undergo mitosis in what will be considered a crowding environment where there are a lot of neighbors and there is relative little rounding up because the cells do not have much space, where in the 2D scenario, even if they have cell-cell adhesion during interphase with their neighbors, they have so much space to round up that they will be kind of individually behaving when they are in mitosis. And so here we confirm that this way of culturing these cells actually makes a huge difference in their cell volume, which is what is expected, and also in their aspect ratio, which is also what is expected, and makes this, even if it's of course not the same, comparable to the differences we were observing in terms of crowding in E13 and E16 brains. And so now we can film these cells, we can see how they behave. You see the two centrosomes are very close by. They make a bipolar spindle as expected, and then the chromosomes divide into daughter cells. This is the same for 2D. Basically, we do not see many differences in terms of centrosome and spindle assembly behavior in terms of timing. But what became evident is that in terms of chromosome segregation, which we are monitoring by the presence of these lagging chromosomes, most likely because of merothelic attachment, things are very different. So here there is a graph with unchallenged conditions where we look at the frequency of cells with lagging chromosomes in either 3D with neighbors or in 2D without neighbors. And you can see that there is always less chromosome segregation depending on the experiment when compared 2D to 3D. If we now challenge mitosis with a spindle assembly checkpoint, MPS1 inhibitor, or with a SEMP-E inhibitor, SEMP-E is a kinesin required for chromosome congression, you will see that this is exactly the same, suggesting that indeed being in a 3D environment with a lot of neighbors makes these cells less capable to segregate chromosomes accurately. We are aware, of course, that this may be contrasting with what has been shown by Christine Naus and Angelica Heyman a few years ago, but I think this is most likely explained by differences in the cell type being studied. So now the question is, how do spatial constraints impact spindle morphogenesis and chromosome segregation? And so we had several hypotheses that I'm not going to be telling you about because they're all negative. We didn't see any differences in volume or mitotic timing or centrosome positioning at mitotic entry that could explain these differences. But I'm going to be focusing on microtubule polymerization activity, and we have to acknowledge that this was partly influenced by the work of Holger Bastian's lab, where he has shown that decreased microtubule assembly rates influence chromosome segregation in colorectal cancer cells. And so we all know that microtubules are quite dynamic and stable, and this instability depends on how microtubule-associated motors associate with polymers and also on how many tubulin dimers are available for growth or depolymerization. And one of the ways of looking at microtubule dynamics is by focusing on EB proteins. Here I'm showing a movie of a EB3 protein, where you can see that this very dynamic behavior being being nucleated from microtubules being nucleated from the poles. And so if we do live imaging analysis and look at the number of EB3 comets per pole, we can see that there is always decreased in cells that are isolated with compares in cells with crowded environment, but also the distance that they travel, it's decreased. Now, if we do the same type of experiments, but before starting measuring EB activity, we actually depolymerize the microtubule cytoskeleton and allow for repolymerization for a very short time window. We can see here 1.5 minutes. We see exactly the same, suggesting indeed that 3D settings where cells are under constraint in a crowded environment have higher microtubule polymerization rates. Now, we wanted, of course, to challenge this. And so what we did is that we increased the polymerization activity using a UMCAC molecule, which is an MCAC agonist, and MCAC is a depolymerizing enzyme. And so here what you can see is that this works because we can have less EB-win comments, and they also reach shorter distances, always in 3D. And importantly, when this treatment is used, we see a decrease in lagging chromosome frequency, either in challenged or unchallenged conditions. We did the opposite in 2D, where we now actually treat these cells with dextran so that they kind of feel that there is a lot of crowding, and we can see that the aspect ratio changes, and so does the behavior of EB1, as you can see over here, and more importantly, the level of chromosome segregation, exactly in the other way around of what I just told you for 3D. So with this, we're learning that apical radioglial cells, at least in the mouse brain, are sensitive to tissue mechanics, and that they respond to high constraints by having high microtubule polymerization rates, which is translated with high chromosome segregation rates. The point here is, what are these cells feeling to change the way they are nucleating microtubules? And so we went back to in vivo and we characterized several things, and one of them was the actin cytoskeleton. And what Vero could see is that the way actin is decorating the cell cortex is actually very different from E13 to E16.5. There's more actin associated with the cell cortex, but also there are these kind of patches making this, what looks like these actin clouds at E13 that are absent at E16. We also saw increase in actin binding proteins, but in the interest of time, I'm not really showing them here. And so if there's more actin at the cortex, could this actually be influencing the way the cortex itself is behaving? And to test this, we go with a very sharp laser so that we can do a very tight laser cut and see the velocity recoiled after the cut. And this is done with the purpose of having measures of relative tension. And you can see that this is very different from E13 and E16, suggesting that the cortex at E13.5 is actually stiffer than at E16.5. And so what we decided to do first is to inhibit actin nucleation at the cell cortex with an inhibitor against ARP23. We can see differences in the way a member of this complex is actually localizing, as you can see over here. And once more, these were impacting the way EB1 was behaving, as you can see by these graphs, but also how chromosomes were missegregating. Would this be also true in vivo? And we think the same mechanism is actually true, because if we now treat cerebral cortex with for very short periods of time with the same inhibitor, these lagging chromosomes that we can identify every now and then become less frequent. The quantification is here, where the mean number of comets and the distance traveled is also decreased. So we're building this idea that the crowding environment of a developing brain, and most likely this is also true in humans, is associated with requirements of having higher levels of cortical actin, which instructs for microtubule polymerization rates that are actually increased when compared to later stages of development, leading to errors in chromosome capture and errors in chromosome missegregation. I will now change completely to another project that's been developed in the lab that is related with the polyploidy paradox, and it's not actually being developed in the brain, even if the initial point of this project was actually developed in the developing Drosophila brain. So as you probably know and are convinced by the first part of this talk, most cells are diploid, meaning that they have these 2 copies of all chromosomes, and there are exceptions to this, such as physiological or scheduled polyploidy, where certain cell types, such as cardiomyocytes, hepatocytes, and megakaryocytes, increase the number of chromosomes they contain, but in a very balanced manner, because all chromosomes here are actually present in multiple copies. And this still quite debatable in the field why certain cells undergo polyploidization, but one of the characteristics that it's typical of polyploidy is an increase in cell size. Now, we are interested in this question, and that hence the title of this slide for polyploid paradox, because if we think about human cancers, we now know that the second most frequent event it's also generated to polyploidization of cells that should not become polyploid. This is normally associated with whole genome duplications that lead to high levels of genetic instability. So the key point is why a certain condition can be advantage and be actually not generating genetic instability, and this exact same condition leads to transformation. And this is one of the questions we have in the lab, and we try to compare both physiological and non-physiological polyploidy to answer to this question. And the project I'm going to be telling you about is being spearheaded by Simon Jembol, a CNRS researcher in the lab, with help from Margot Boudzik, Anthony Simon, and Corentin Vertel. And so a few years ago, when Simon was starting to work on polyploidy, he developed all these systems that are chemical or genetical, where RP1 cells or HCT116 or BG fibroblasts can be manipulated to generate tetraploid cells, either to cytokinesis failure, mitotic slippage, or endoreplication. The differences between them is that in cytokinesis failure, there will be 2 nuclei, mitotic slippage 1, and endoreplication 1. But something very curious Simon noticed was that when he used techniques to induce mitotic slippage, he saw that in contrast to this beautiful, very spherical G1 diploid nucleus, the G1 tetraploid nucleus upon mitotic slippage was quite deformed. And so we wanted to understand how mitotic slippage leads to nuclear defects. And so here I'm showing you a nucleus that is going into mitosis. You can see 2 daughter nuclei, and if you compare the mother nuclei with the 2 daughter nuclei, they seem very similar, except that they have, of course, different sizes. Now, if we induce mitotic slippage, and this can be achieved either through chemical inhibition of EG5 and MPS1, or using anoxin anoxin degradation for cyclin B, which is fused to an AID, we will obtain these very deformed nuclei that look like even a mouse embryo, as someone pointed out this to me the other day. And so the key point is, independently of the way we are generating mitotic slippage, we are obtaining these deformed nuclei, and we wanted to understand why. So we're going to show you this, I'm going to show you this kind of measures throughout my talk, where nuclear circularity is going to be measured to show how close to one, to a circle, these nuclei are. And you can see that a cell that undergoes mitotic slippage has very low levels. And nuclear solidity to measure the levels of invagination, where you can see that they're also quite low in cells that undergo mitosis compared to mitotic slippage cells. The choice of using instability, and so from one to many in the title, comes from the fact that there's almost like not a single nucleus that will resemble another one upon mitotic slippage. So here on the left you have diploid cells, and with the dashed lines the nuclear shapes, and they are quite comparable. But if you look upon mitotic slippage, you can see that these are very variable. And this is, again, not related with whole genome duplication, because if we induce cytokinesis failure or endoreplication, nuclear shapes are very stable. This is true for RP1 cells in a Petri dish, but is also true in vivo. So if we use megakaryocytes as a model system to study physiological or programmed polyploidy, and we chose those because they are formed through mitotic slippage, they have this very abnormal nuclei, and we actually learn how to purify them by isolation of hematopoietic diploid progenitors that then undergo differentiation and form these megakaryocytes. And I'm sharing here this movie because you can see that megakaryocytes are formed by mitotic slippage. This is actually something that it's still debated in the field. You have this nucleus here, now the chromosomes condense, and then there is chromosome segregation, but they fall back and form this single nucleus, and we do not see any furrow ingression by phase contrast, or this cell where you can see that chromosomes start to condense and then they exit without any anaphase movement, suggesting that indeed megakaryocytes undergo polyploidization through mitotic slippage. Now, if we compare the shape of this nuclei with other cells, that are also polyploid, you can see that, again, once more, the instability is quite obvious. These are smaller megakaryocytes where we see already differences in circularity and solidity, and they become much more prominent in larger megakaryocytes, where in hepatocytes, in salivary glands, this is really not the case. And hepatocytes are generated through cytokinesis failure, and salivary glands through endoreplication. So we wanted now to go back to the origins of these nuclear deformations, and Simon decided to film these cells using several markers. And when he used a tubulin reporter, he realized that there was a lot of microtubules associated with the invagination regions, as you can see over here, or under these tills. And so we decided to remove the microtubule cytoskeleton with a very short depolymerization using no-codazole. And you can see that this actually improves the way these mitotic slippage nuclei look like, as you can see over here, both in terms of circularity and solidity. During these movies, we also had the impression that the nuclei were actually more soft. And so in collaboration with Sarah Wickstrom and using atomic force microscopy, nuclear stiffness was measured. And you can see that there are differences independent of the cell line used that show that tetraploid nuclei are actually more softer than diploid nuclei. To see if we can actually recover this behavior, Simon incubated these cells with histone acetylase inhibitor, and he can see that now this lead to an improvement in the way these nuclei actually look like compared to controls. Now, we wanted to have an idea if these differences in nuclear compaction, or at least in nuclear behavior, were actually informing us on maybe there will be differences that could be explained by nuclear organization. And so Simon set up this method where after induction of mitotic slippage, diploid and tetraploid cells can be separated through FACS, taking into consideration the stage of the cell cycle and DNA content. And with the help of Elisa Riccio and Giovanni Cirello, we look at chromosome organization. And we were surprised, although it makes sense in light of previous data obtained by Elisa's lab, where we can find more contacts between short and long arms, but we did not find huge differences in the way the chromatin domains are actually set up between tetraploid and diploid cells. I'm not sure I actually said that, but we are looking at nuclear organization exactly just after cells undergo mitotic slippage, so in the first G1. And so to try to understand where how these softer nuclei are actually assembled by failing mitosis, Simon decided to perform a mini screen using super resolution microscopy to compare nuclear envelope markers and chromatin markers between diploid and tetraploid cells, taking into consideration the fluorescence in the whole nucleus or around the nuclear envelope, or in regions that are intact or deformed. And I'm not going to go through all this data, just to highlight that there were very interesting differences found with lamin C and with H3K9 dimethyl. And so these differences here can be seen in this diagram, where you can see that places of invagination, there are less, in places of deformation, there are less less H3K9 dimethyl and also less lamin C, and the quantifications are over here. What is interesting also is that in regions that have low lamin C, we can see a compensation by having high lamin B1, which has already been reported in other cell types. Now, with the help of Geneviève Almosny, we did ChIP-seq for H3K9 dimethyl and also H3K9 trimethyl, but I'm not going to show this. But what I can show you here, that indeed the distribution seems to be affected with a lot of enriched peaks typical of tetraploid cells that we're not seeing in diploid cells throughout the genome, suggesting that indeed H3K9 dimethyl is not distributed equally between diploid and tetraploid cells. But what is H3K9 dimethyl? Actually, H3K9 dimethyl is considered to be a mitotic guidepost that can allow in certain cells cell identity to be reestablished after mitosis. And in very interesting work published in the last years, it has been shown that upon nuclear envelope breakdown, the phosphorylation of H3K9 serine 10, which is the residue that next to the K9, so H3S10 phosphorylation, sorry, I said the lysine, I shouldn't, which is serine, which is localized just next to the lysine. Its phosphorylation will allow the dissociation between chromatin and lamins, and upon dephosphorylation of this residue, the chromatin and laminin interaction can now occur. So it has been postulated and shown by many different labs that H3 serine 10 is phosphorylated by Aurora B. We tried quite hard to see if we can inhibit Aurora B, but just the inhibition of Aurora B actually led to mitotic slippage, and so we were not able to see if this had an effect on the behavior of our cells that were also undergoing mitotic slippage. So we tried to consider what could be actually going wrong if cells exit mitosis with high levels of phosphorylation. This is how mitosis actually should look like, where H3K9 dimethyl levels will be quite stable, but not phosphorylation levels. So if now the cells exit normally, phosphatase activity has occurred, and so there are no phosphorylation levels. But if they exit mitosis prematurely, which is the case of mitotic bypass, mitotic slippage, then these cells will exit mitosis with high levels of phosphorylation on serine 10. And so to challenge this possibility, this working hypothesis, we decided to take into consideration this concept of the phosphorylation and the methylation had to be actually mutually exclusive. And so we treated diploid RP1 cells with histone demethylase inhibitor called methylstat, and we can see that indeed there are less, the levels of phosphorylation decrease. And to go a step further, we now express in these cells H3K9, a mutant for phosphorylation on replacement by alanine, H3S10. And this, as you can see over here, even if it doesn't improve completely, actually improves substantially when compared to controls. If you look at the nuclei, also you can see the differences between tetraploid nuclei as control or the ones where cells are expressing the alanine mutant. Now, we also treated these nuclei with methylstat, and you can see that indeed this actually leads to an improvement not only of H3K9 dimethyl signals, but also of lamine ratios. So the final proof came by inhibiting phosphatase at mitotic exit and see that in diploid cells we have high levels of serine 10 phosphorylation and these nuclei that are highly deformed. So we're building this view that mitotic slippage actually generates cells with these problems in serine 10 phosphorylation that do not allow the reestablishment of chromatin and lamine interactions. Now, does this have an effect? I showed you before that in the first G1, there were not differences at the high C level of the whole population. There are a few already by RNA-seq, but these actually become much more evident in the first G2, suggesting that indeed these differences in chromatin and laminin interaction will probably lead to differences in gene expression that will be then leading to gene expression alterations. So with this we conclude that exiting mitosis prematurely would lead to softer nuclei and invaginations imposed by the microtubule cytoskeleton, which allow mitotic exit to occur with high levels of serine 10 phosphorylation, inhibiting chromatin and nuclear laminin interaction. Now, approaching the end, could be that the mechanisms revealed in RP1 cells are contributing to the nuclear deformations in megakaryocytes, which are, to our knowledge, the only cells that in vivo undergo mitotic slippage. And so first we look at the microtubule cytoskeleton, and indeed we see that regions of nuclear deformations, we can see microtubules associated with these regions. Now, we were very lucky because in the literature we could find already that megakaryocytes have softer nuclei when compared to their progenitors. But more interesting, we were able to see differences in H3K9 dimethyl, also in regions of deformation, as you can see over here and on these quantifications. And importantly, if we inhibit histone demethylases, we now see that the nuclear of these small megakaryocytes, at least, is actually improved. So the outlook of this part will be that inducing whole genome duplication through mitotic slippage actually has different consequences of inducing whole genome duplication through other means. That leads to softer nuclei that can be deformed by the microtubule cytoskeleton, to nuclear morphology defects, and this nuclear instability that we think can actually be a characteristic of at least megakaryocytes. And with this, I leave you with this slide. I'll be very happy to answer to questions. And we rely on a lot of collaborations that I have mentioned and many more that I have not mentioned that are not relevant to this work, but relevant to other work that we are developing here in the lab. Thank you for your attention. I will stop sharing.
Thank you very much, Renata, for this very inspiring lecture. So just a reminder to the audience, so you can use the Q&A to ask your questions, or I can also unmute your microphone. So maybe I can just ask the first question while people type questions into the chat. I was just wondering, so for the megakaryocytes, is there any evidence for more genomic instability as compared to other hematopoietic cell lineages, or as compared to cells that don't show any mitotic slippage?
So we have not investigated this question, but I know that Beth Patsila in Oxford is actually looking at this exact question. So I think very soon there will be some very interesting data related to this question.
Thanks a lot. I can't see any question yet, so maybe I just ask another one to the first part. So regarding your MK agonist for the first part of the talk and then the microcephaly, so do you see any clinical potential for this UMK agonist, in terms of decreasing misegregation frequency in patients with microcephaly?
Actually, I'm not sure this will be possible to be used because the brain is, mostly assembled in utero, and so I will think that this will be very toxic given in utero, as one could imagine. Afterwards, I also think might be quite dangerous because it will maybe increase genetic instability or chromosome instability in other tissues. I think the point here maybe that is important to take is that the levels of chromosome instability that are normally associated with cancer, or even this behavior that it's normally associated in cancer, it seems to actually also be present during neurodevelopment. And so this brings to a question that I think is also quite relevant is, could it be that the way you are able to be tolerant to this type of chromosome instability is different in utero than in adult tissues?
Thanks. There is one question from Dilaka Welapili in the chat. So have you seen the tetraploid cells stably continue dividing while staying as tetraploid? Or do they become diploid after a few divisions?
So it's true that they then undergo multipolar divisions as high very frequently, and this will generate not tetraploid daughters. But in our hands, we don't see, at least with the type of analysis we do, we don't see them becoming diploid. They will be not fully tetraploid, but they will have chromosomes content of more than 50 chromosomes, but not 90 and a lot.
Thanks a lot. I can't see another question yet, so then I'll just ask one more to the first part. So for the microcephaly, do you see similar effects on all cell lineages in the brain, or do you have a bias for neurons or astrocytes or oligos in terms of numbers?
Yeah. So we have not really done this analysis, but we think basically at the stages. So the initial work in mouse, we generated a mouse model with centrosome amplification. And in this mouse model that had centrosome amplification in neurostem cells or in apical radioglial cells, we saw that there were neurons that had nuclei that do not have the same size as other neurons, but we did not really extend, so they would be aneuploid. We did not really extend this to other cell types. But I think our work in flies, which was inducing aneuploidy, we could see differentiation towards a neuronal fate or a glial fate, both.
Okay.
So I think this is also possible.
Thanks a lot. There is one more question in the chat. So did you do any transcriptomic or proteomic analysis in tetraploid cells compared to the diploid ones?
Yes, maybe I went too fast towards the end. We did. On the first cell cycle after becoming tetraploid, we compared diploid and tetraploid in G1 and diploid and tetraploid in G2. And we saw that in G1, there were not major differences, but there were a lot of differences in G2. Not a lot, there were more differences. There were around 400 genes. And so what we think is that there is indeed some differences, at least of gene expression, but probably according to what will happen afterwards. So these cells will enter mitosis and may continue to proliferate or not. They will maybe become more homogeneous in terms of gene alteration. I don't think this is that 5 cell cycles after the same gene expression alterations will be found. Maybe this is a way of making it more clear.
Thanks. There is one question from Nalini Kodas Srinivas. A general question: in cells already with low lamin A, lamin C expression that continue to proliferate, how is the mitotic slippage affected?
So we haven't looked at cells if they do continuously mitotic slippage, with the exception of megakaryocytes. In our setup in human cells, in our P1 cells, we induce mitotic slippage once, and then they will exit, they enter in the cell cycle, they enter mitosis, but then they will not undergo mitotic slippage again. So we haven't looked in repeated mitotic slippage. In megakaryocytes, they do, but it's also different in these cell types because even the levels of expression of lamin B1 and lamin AC changed during differentiation. So I think it's more a process of the differentiation itself than of the level of ploidy and having failed mitotic slippage several times. To do these experiments in our hands, we will have to inhibit p53 or deplete p53 because otherwise they will tend to arrest in the cell cycle.
Thanks a lot. I cannot see any more questions in the chat, so thank you again, Renata, for this fantastic.
Okay.
And yeah, so our next lecture will be after the summer break on the 17th of October, as mentioned in the beginning. So thank you very much, everyone, for joining, and enjoy the rest of the day.
Okay, thank you. Bye-bye.
Bye.